Hello, everyone, and welcome to today's live broadcast. Seeing through the Murine heart, quantitative 3D assessment of myocardial ischemia, reperfusion injury and response. Presented by Simon Merz, Institute for Experimental Immunology and Imaging, Essen, Germany. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Imaris. For more information on our sponsor, please visit imaris.com. Now let's get started. I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab down at the top right of the presentation window, or let us know you're experiencing a problem by using the Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce our speaker, Simon Merz, Institute for Experimental Immunology and Imaging, Essen, Germany. And joining us for our live Q&A, we have Sebastian Costa, University Hospital, Essen, Germany. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Simon, you may now begin your presentation.
Hello everybody, and special thanks to Imaris supporting this broadcast, and welcome to the webinar. My name is Simon. I am a third-year PhD in the Institute for Experimental Immunology and Imaging in Essen, Germany. Today, I would like to talk about how we came to see through the murine heart. If you would like to read more about the topic, the paper was published recently in Nature Communications. This project was a collaboration between us and the Department of Cardiology and Vascular Medicine in the University Hospital, Essen. But let's start at the beginning. So why do we actually want to see through the murine heart? What goal do we hope to reach? First of all, why do we look at the murine heart at all? So the background here is myocardial infarction, which is still a very, very lethal disease, accountable for over 9% of all deaths in the EU 28 in 2015. This is more or less true around the globe, so a lot of people suffer from myocardial infarction each year. Therefore, a lot of research is conducted to understand the biology behind myocardial infarction, to investigate the associated immunological response in order to come up with new drugs eventually. In this project, we worked with a mouse model close to the clinical reality, an ischemia reperfusion model. Here, in the living animal, one coronary artery is occluded via a knot that is pulled tight for 45 minutes, inducing ischemia downstream in the muscle, which is no longer perfused. After 45 minutes, the knot is loosened and reperfusion is allowed for 24 hours or longer times, similar to placing a stent. After this time, the heart is then excised and analyzed. This is when you are exactly facing the readout problem. What do you actually want to know? Because you have to decide. If you want to know about infarction parameters, like which regions are damaged, which were still perfused during ischemia, etc., the heart is typically sliced into 2 mm thick slices for a redox TDC assay. I'll explain that in more detail later. However, besides giving you information about the extent of the damage, this approach yields no information on cellular identity or localization whatsoever. Therefore, you can cut the heart or your TDC slice a bit more and do ordinary two-dimensional sectioning, which gives you then limited information on cellular identity, location, and quantity, but very limited to a two-dimensional space. Or you take the entire heart, dissociate it completely, and do flow cytometry. This will give you a quantitative overview of your successfully prepared and stained cell populations without any localization context at all. So if you are in need of all this information together, you have to have multiple animal groups, introducing you a lot of variability in the testing. Therefore, we wanted to establish a quantitative whole organ multiplex imaging approach assessing myocardial infarction. That brings us to the question, how do we actually look inside an opaque organ? So let's assume you have treated the mice with drug that you want to test. You can also inject fluorophore conjugated antibodies IV into the living mouse shortly before excising the heart. The heart itself, even after thorough perfusion, is quite reddish, what you can see on the lower left side now. This changes into gray once the heart is chemically fixed. Next, we remove all the water from the sample by dehydrating it in an ascending ethanol series. With all the water gone, we can match the refractive index of the sample with ethyl cinnamate, a non-toxic organic solvent. This minimizes scattering of light as it passes through the sample, rendering the sample transparent. However, the heart muscle is still densely packed with residual erythrocytes or pigments like myoglobin, which block the light's way. And these endogenous substances are not only blocking the light, they are also very autofluorescent. So in this example, we illuminated a cleared whole heart from the side and took an overview image. Longer wavelengths can penetrate deeper into tissue. So the signal intensity from right to left, as you see here in the histogram, is not severely dropping. We also say the redder, the better. However, this is quite different when assessing tissue autofluorescence with shorter excitation wavelength, like 561 nanometers here in the middle in gray, or even much worse, using 488 nanometers on the left in cyan. Using linear display, we don't even see the inner muscle, only a bright halo of autofluorescence at the edge of the tissue. Having such gradients in illumination and emission intensity throughout the sample prohibits any approach for quantitative assessment. Therefore, we compared different bleaching protocols which destroy pigments and autofluorescent molecule within the sample, so that we get a better overview. We tried pseudo-black bleaching, which turned the sample completely black, rendering whole mount imaging absolutely impossible. What works for brain slices does not seem to work in a whole organ approach. With heme illusion, the bleaching step of the cubic protocol, which is another clearing approach using aqueous solutions, the sample became much clearer. However, we observed spots in the muscle tissue and higher autofluorescence in the channels we actually wanted to use for quantification once we cleared the heart in ECI. Therefore, in contrast, the published balance approach is capable of homogenizing the tissue's autofluorescence and results in transparent samples. Furthermore, it is fluorophore compatible, so IV injected fluorophore coupled antibodies can still be detected afterwards. So with this bleaching approach, hearts become white and, once immersed in ECI, rapidly cleared, as you see in this time-lapse video spanning about 30 minutes. We termed the process balance, which stands for bleaching-augmented, solvent-based, non-toxic clearing. Balance, as I showed you before, and this completes the picture, lowers therefore and homogenizes tissue autofluorescence in all channels, which is a prerequisite for quantitative imaging. So to answer the initial question that we asked, we can look inside an opaque organ using a pigment bleaching step for homogeneous imaging coupled with tissue clearing. Now that we have the organ clear, we use light sheet fluorescent microscopy, in short LSFM, to image an entire organ with single cell resolution. So in LSFM, we apply an up to 4 micrometer thin sheet of light perpendicular to the objective. This planar illumination leads to very, very selective excitation of the sample, reducing photobleaching. We also acquire the whole field of view simultaneously, which reduces acquisition time. The sample itself, as you see here in this cartoon on the right-hand side, is then moved through the applied laser sheet. And this is how we get the fast recordings of image stacks consisting of 2-dimensional optical slices that can quite easily be turned into 3-dimensional reconstructions by the use of appropriate software like, for example, Merz. What you see now in the lower left is such a stack of 2-dimensional optical slices of the aortic valve. We are viewing it from above, looking into the left ventricle. In both 2-dimensional and 3-dimensional representations, you can appreciate the fine structure of the 3 valve cusps. Also take note of the detail of the vessels that surround the valve. So now we're looking at the entire heart, and we always image the hearts as one, no cutting involved. Here you see a three-dimensional reconstruction of the dorsal side of the murine heart based on autofluorescence and CD31 staining, which is an endothelial marker. We can clearly distinguish the right and the left ventricle and the muscle walls. The appendages and the aorta with the aortic valve are also visible. So if we have a closer look on the left ventricular wall, we see a homogeneous autofluorescent signal throughout the muscle, yielding enough contrast to distinguish even single cells, most likely cardiomyocytes. And if you have a very close look, you can even spot dimmer autofluorescent regions that might represent the nuclei. Also, with bleaching, you see still some spots of high autofluorescence left, and in the enlargement below you see that these spots also attribute to certain single cells, but we don't know what causes it yet. However, in general, we have a good overview, and therefore with bleaching, clearing, and imaging technicalities established, we started to bring myocardial infarction parameters to three-dimensional level. Since we want to establish a multiplex approach, we need to determine what we are able to visualize simultaneously so that LSFM is capable of answering multiple questions alone. Typically, as mentioned before, cardiologists also and also other researchers studying ischemia reperfusion perform the TDC assay. A heart is there sliced into thick sections in which three major zones can be distinguished. The remote zone is the part of the heart muscle that was still perfused during ligation. To label it blue, as you see here, the coronary artery that was occluded to induce ischemia in the first place is religated, so closed, again before cutting the heart into slices, and Evans blue, a dye, is pressed through the aorta, staining everything that is perfused. The TDC assay then itself is a redox reaction on the fresh tissue. So if cells are or were metabolically active during excision of the heart, TDC becomes red. So the part of the red tissue that is Evans blue negative is termed the area at risk. This area was not perfused during artery occlusion, but most cells survived and are still metabolically active. Within this zone lies the real ischemia reperfusion injury, in which TDC cannot be reduced to its red form, marking the damaged or dead cells white. So, having understood this, and in order to reproduce these parameters in 3D, we performed the ordinary ischemia reperfusion challenge, excised the heart, religated the artery, and filled the heart with Fitz albumin instead of Evans blue. This led to an easily distinguishable perfused remote zone, which is FITC positive, and a FITC negative non-perfused zone, the area, or better in this case volume, at risk. Note that the area at risk is below the knot where we actually suspect it to be. The total heart muscle volume was obtained by segmenting tissue autofluorescence alone. And this is always much better to view in a video. So this video shows this exact same thing in 3 dimensions using Merz. So a reconstruction of the tissue's autofluorescence in gray gives us a representation of the entire heart muscle. So on one side you can spot the knot used to occlude the artery. The corresponding segmentation is white. With such a 3-dimensional reconstruction, we can now look at the muscle tissue layer by layer in a 3-dimensional environment. Note that we also see some autofluorescent spots in the tissue even after bleaching. The FITC signal is shown in cyan now, and we clearly see the FITS positive remote zone and the non-perfused area at risk, which is FITC negative. In this 3-dimensional segmentation now of the autofluorescence, here in gray, and the area at risk, shown in orange, now allows us to quantify the heart muscle volume as well as the volume and relative location of the area at risk. In summary, using just 2 channels, FITC and tissue autofluorescence, in 561 nanometer excitations, we are able to simultaneously quantify the area at risk and the heart muscle volume respectively. But what about the damage? So here, a paper from 2017 described impaired mitral vasculature in stroke brains. And therefore, we thought that we could take a look at the vasculature in the murine heart as well, using, as I said before, CD31, also known as PCAM1, as a marker for the endothelium. To investigate the basal state, we injected fluorophore-coupled anti-CD31 antibodies IV into a control mouse prior to the excision of the heart. And this is what we found: very, very densely packed, homogeneously distributed blood vessels. And what you see that here is one single optical slice on the left, and the small three-dimensional reconstructions of the different muscle walls show a very structured vascular network as expected on the right. Having such an orderly vessel meshwork in basal, untreated hearts, we were interested in quantifying the system's complexity in 3D now. So therefore we used a filament model approach, and we found that there is a total vessel length density of 2.2 meters per cubic millimeter. So it's a lot of cable basically within the left ventricular wall. And this is about twice as much as in the brain. So as a measure of complexity, we determined the vessel length between two branch points to be 107 micrometers on average. Knowing this, we looked at the IR injury hearts after 24 hours of reperfusion. And here we found a hole in the microvasculature, while vessels outside of the CD31 negative zone are still arranged in an orderly fashion. Taking a closer look, we find that CD31 positivity is mainly lost in smaller vessels, while the larger ones survive. This matches also with the findings in the brain paper as well. However, CD31 negativity is the absolute lack of any staining compared to CD31 dim regions, which are enlarged in the upper right here. So interestingly, these match with highly autofluorescent, randomly shaped and distributed regions of yet unknown identity. Anyway, we used the border between CD31 positive and negative microvessels to trace and thus quantify the IR injury. This brings us back on track. So on average, the IR injury assessed by CD31 negativity takes up about 4% of the total murine heart. When we looked at the hearts after 5 days of reperfusion, we again encountered a changed pattern. So the size of the IR injury was drastically decreased. The CD31 negative region was surrounded by seemingly chaotic CD31 positive vessels, so we termed that region CD31 curly. It is the area within the dashed white line here. Looking at the three-dimensional structure of this zone, we found a reduced vessel length density, but a doubled complexity. Interestingly, we found that the volume of the CD31 curly region amounts approximately to the size of the IR injury after 24 hours of reperfusion, giving us, technically speaking, a proxy of where the damage has been 4 days before. And this is, of course, very interesting when we look at immune cells later. So we see a loss of CD31 staining within blood vessels, but does that have anything to do with the actual damage that has been assessed for decades? To address this, we challenged mice with ischemia reperfusion, injected fluorophore-coupled antibodies, anti-CD31, IV into mice 10 minutes before excision, sliced the perfused hearts and performed the TDC redox staining, and afterwards we took the same slices and cleared them to do correlative LSFM using CD31 as a marker. Here we see what the TDC slices of an exemplary heart look like, with the white zone being the damaged tissue regions. When assessing this in light sheet fluorescence microscopy, using CD31 as a marker, we found that CD31 negative zones overlapped with TDC negative tissue areas. This is underlined by the good correlation of TDC negative and CD31 negative regions per slice in percent. However, TDC planimetry is done by slightly compressing the tissue slice with a glass slide, taking the image from above. Using light sheet fluorescence microscopy, the loss of CD31 does not seem to be severe when looking at the maximum intensity projection alone. However, the pattern becomes very distinguishable when browsing through the optical slices one at a time. And when viewed from above, the three-dimensional segmented CD31 negative volume, here in yellow, looks quite familiar when you look back to the TDC enlargement. But the true three-dimensional complexity of the volume in Z can only be assessed by light sheet fluorescence microscopy. The IR injury itself, measured via CD31 negativity, also correlates well with the mouse heart's ejection fraction reduction after ischemia reperfusion. This was also measured within the same hearts, so allowing multiplexing of physiological and light sheet fluorescence microscopy readouts and benchmarking against established methods. But enough images, let's watch some videos, because this is why I love light sheet fluorescence microscopy, since it conveys three-dimensional information much better than any explanation. Here we see CD31 in green and autofluorescence in gray. The knot is again segmented and displayed in white. We can appreciate the CD31 negative area, so basically the hole in CD31 staining, and this is traceable and can be three-dimensionally quantified, what you can see here in yellow. Furthermore, CD31 labeling also allows us to trace the heart's major arteries, shown in yellow, and veins, shown in blue. By simultaneously tracing the knot, we are able to deduct which arteries have been occluded during the operation, shown here in red in a moment. And this basically gives us the physiological area at risk. Overlaying the traced one in orange here, we see that the two are matching nicely. However, overlaying the IR injury gives us a very interesting result. Only the ventral part of the heart is injured, while the other side shows no signs of damage. Identifying these specific regions of interest and unlocking them for further deeper analysis is one of the main advantages of this system. So what can we do now? So we can add IR injury quantification via CD31 and reconstruction of the vessel tree to the list of what we can visualize simultaneously in one murine heart. Since we can now reconstruct the IR injury in 3 dimensions, we went ahead and applied a clinically accepted heart segmentation normally used for human tomographic heart data to our LSFM datasets. Hereby, the heart is divided into 17 segments. By scoring how often IR injury was located within a certain segment, we created a heat map to visualize our findings in mice in a clinically accepted way. We can clearly see that the volume covered by the IR injury is shrinking when the time of reperfusion is prolonged, going to the right side. The extent of the CD31 curly region after 5 days of reperfusion is larger than the actual IR injury measured at that time, which was also expected. So now we also looked at the location of immune cells. So here you see CD31 in green and the big hole in the microvasculature after 24 hours of reperfusion. While we fly through, we see now neutrophils in cyan, and you can already see from these reconstructed raw data that the cells are lining the IR injury, but not penetrating into the damaged tissue. Using Merz then, we were able to segment and thus count neutrophils in the entire heart. So in combination with the myocardial infarction parameters that we assessed before, we can now group the cells according to their location. Interestingly, we'll find in this heart that only about 1/3 of all the cells are located at the border of the IR injury, while a substantial amount of cells also reside in the remote zone. So after 5 days of reperfusion, we see a very different picture here. So the IR injury is barely visible, and the surrounding CD31 curly region has a slightly brighter signal compared to the orderly structured microvessels around it. Also, the presence of neutrophils here is strongly reduced, which also matches the FACS data. This can also be seen in the segmentation. The few neutrophils are located around the IR injury in yellow, which consists only of multiple small volumes. It is surrounded by the CD31 curly volume in green, in which about 40% of all neutrophils reside. Summing it up, we now have a tool multiplexing immune cells and infarction parameters that is also not restricted to the murine heart. And this allows us to identify subpopulations of responding cells or research their overall location. So what we can add to our list is that we can now show immune cells simultaneously with all the other parameters assessed before, and that the data can be presented in a clinically accepted way. Besides these points, the light chain fluorescent microscopy approach, lasting about 1.5 days, is quite fast compared to all the other methods you would have to do to obtain the same result. Multiplexing readouts from one animal also fulfills the 3R criteria. And above all else, you can explore your data. Find regions that behave differently than expected. For example, the damage we saw in the beginning that is not homogeneously distributed. Or you can study the distribution of immune cells and their involvement in such phenomena. Or, seen from a very, very technical point of view, you can check if the knot actually hit the coronary artery when you happen to wonder why there are no neutrophils present in your sample. So with the flow cytometry, as an example, you just had to brand the data as an outlier without exactly knowing why. After having shown you what three-dimensional microscopy can do, let me close with a story from our lab. So when we submitted the paper for the first time, we went dining in a Chinese restaurant. And after dinner, I was presented with a Chinese fortune cookie that actually read, quite fittingly, "Whatever you do with your entire heart will bring the best results." And with that, I want to close and thank all the people involved in this project, mainly the supervising PIs and the hardworking PhDs with whom I had the pleasure to work on this project. So I thank you, of course, for your attention.
Thank you, Simon, for that excellent presentation. We will now start the live Q&A portion of our webinar. Now, if you have a question you'd like to ask Simon and Sebastian, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. Our first question: is the balance protocol restricted to the murine heart?
Yeah, so that's a good question. No, it is not. So you can process a whole different kind of organs with the balance protocol. For example, in the paper we showed that you can process nicely the very heavily autofluorescent organ, the liver, and just by autofluorescence display the vessels inside the liver. But this is not even restricted to the mouse in general.
We also did the human heart samples that we got from biopsies, and it was a pretty good clearing effect at the end. It's a bit different for the mouse in concerning the staining part, but overall the protocol for the clearing is the same. And I think we also did a few other organs like lung, brain, and kidney. I mean, if you do the protocol like we did with the intravenous injection of the antibody, you basically stain the whole mouse with your desired antibody, so you can process every organ you like.
Thank you, gentlemen. Our next question: how do you distinguish arteries and veins? Just by the morphology?
So we distinguish them by morphology and where they are located. So veins are normally located on the outside of the heart muscle, whereas the arteries are not. But also veins are CD31, let's say, dim, so they don't express as much CD31 as arteries do. And that's why it's kind of easy to distinguish between them. It's sometimes even a little bit difficult to see veins, but especially when the vessels are very big, then you can easily distinguish them.
Also, it's a signal intensity thing. Like the bigger arteries, especially the left coronary artery, is very bright concerning CD31 signal, and the veins are a little denser. And the more you go to the smaller vessels, the less CD31 positive signal you get.
Okay, Simon, I believe this question's for you. Does the balance protocol allow visualization of endogenously expressed fluorophores?
Yeah, the direct answer would be no. So by bleaching the heart, the murine heart, the endogenously expressed fluorophores are also bleached, and therefore their fluorescence is quenched. And we have in the paper, so we tested, for example, the cubic protocol, which is meant to preserve endogenous fluorescence, and that works also nicely on hearts. It doesn't get the hearts as clear as the balanced approach, and it nicely shows the endogenous fluorescence. However, it obliterated the Alexa fluors we are using as the artificial fluorophores. So that was not an option, but that is something that you can do if you are focused on the endogenous fluorescence only. We also tested the normal ECI protocol without bleaching at all, because ECI is also meant to preserve the endogenous fluorescence. However, in the heart with a lot of autofluorescent background, the signal of the endogenous fluorescence was not very bright. And you could only see it in regions where there was not so much autofluorescence or muscle tissue in the background. But it was a nice way of showing that the IV stain that we gave actually marked all the cells that we also could identify in the via endogenous fluorescence. So that's supplemental figure 2.
Now, Sebastian, you use CD31 negativity for assessment of the IR injury. However, CD31 is only an endothelial marker. Is it possible to stain the actual tissue damage?
Well, that's true, but CD31 only stains the endothelium and therefore only the vasculature of the heart. And unfortunately, no, to my knowledge, we don't have a way to image the tissue damage itself of the heart yet. Of course, we're working on it. We have a few suggestions that we are currently testing in our experiments, and we are very hopeful that in the next couple of months we will get something to stain the tissue damage of the heart after myocardial infarction.
Thank you. Now we have a question coming in from Thai in Malaysia. What kind of fluorophore was used for this study?
So we used FIT-C for basically marking everything that was, or FIT-C gelatin better for marking the remote zone, basically everything that was perfused during the infarction, and that worked well, also with the bleaching protocol. And else we only used Alexa fluors, so mainly the Alexa fluor 647 and also 790. But these are the ones that we tested since our light sheet system is also restricting us. We have single wavelength lasers, and therefore we can only test so much of the antibodies, or the fluorophores. But what we also tested was Alexa fluor 546 recently, and that also worked well.
Now we have some great questions coming in. I want to remind our audience, for those questions we do not have time to answer today, they will be answered by our speakers via the contact information you provided at the time of registration. But we're going to continue with our live Q&A. Now we have a question coming in from Mike. Do you know if CD31 expression itself can be altered by the ischemia?
Well, we know from the literature that there are a lot of studies done on CD31 expression using standard histology analysis in like the 90s and 80s, mostly in dogs and rabbits, to my knowledge, and also for longer periods of reperfusion when we did. What we showed you today are at the maximum 5 days of reperfusion, and our usual analysis we do after 24 hours, so 1 day of reperfusion. in the longer periods that were published earlier, like that was like 7 days, 14 days, CD31 expression increases in the former ischemic zone, so the damaged tissue, which makes sense as it is revasculated and needs to have blood supply again. We also kind of found this when we did the analysis at 5 days. This like curly area is very bright in CD31 expression. Yeah, so I hope this answers the question.
Thank you. Now our next question coming in from Bill, Northwestern Medicine. Do the curly CD31 regions represent granulation tissue?
Yeah, it will also be a question for me. Simply said, we don't know yet. Granulation tissue is, to my knowledge, analyzed via hematoxylin and eosine staining, and you have a lot of granulates. And we don't see that in the light sheet. So we see from the autofluorescence the overall structure of the of a tissue, but we didn't see a correlating image that we see in histology. So simply said to that question, we don't know yet.
Thank you. Quite a global audience we have today. We've got a question coming in from France. Dario is asking, which modules of Imaris have been used to obtain the different 3D regions and for the vascular system?
So which modules we use from the Imaris software, you mean? Simon, I think we only did what is shipped with the regular software, right?
Yeah. So we use the spot function, for example, to determine where the cells are, and we also use the filament tracing tool to basically determine the vessel length density, etc. Yeah, and I mean the general tracing tools to determine the surfaces. So we also, some surfaces like the FIT-C, we could determine sometimes by the algorithms that are coming with Imaris. But for the most part, we use the tracing tool to do it manually or semi-manual. So sometimes you have a semi-manual tool where you can basically draw regions half automatically.
Okay, how is the probability analysis of the injury done manually?
Yeah, that's referring, I guess, to the 17-segment model. And yeah, that was manual. So we just looked at, so we got a surface in Adobe Illustrator and looked at the specific regions. If there was infarction, for example, we gave it a 1. If there was no infarction, we gave it a 0. And then afterwards, we did the probability over all hearts analyzed.
Thank you. Now, in order to identify leukocytes, do you need to have them labeled beforehand, or can you get antibodies to stain once you are clear?
So what we did in general here is to inject the antibodies IV into the mouse just before the mice were sacrificed. So in that case, you distribute the antibodies throughout the mouse without having to do whole mount staining or something like that. And therefore, yeah, we just did a live incubation, if you want it that way.
Okay, our next question, have you had problems with limited penetration of antibodies inside the tissue? And if yes, how did you overcome this?
So that's a good question. Actually, we didn't observe like, let's say, a penetration border or something like that, and not really, to be honest. We didn't observe a higher background, a fluorescent background barrier or something within the tissue. So I wouldn't say that we have a penetration problem within the tissue.
Yeah, we also once stained a mouse that had an endogenous expression of fluorophore for neutrophils. And when we co-stained with Ly6G antibodies, so also neutrophils, and we found like a 100% match of the endogenous signal and the fluorophore signal. So we are pretty confident to say that the route we used for administration of the antibody labels all cells we want to see, at least for neutrophils.
Thank you, gentlemen. Now let's go to this question coming in from University of Georgia. What other immune cells can you detect?
Yeah, that's also something that we are working on. So in the paper, there was also a hint on macrophages. And of course, we also are interested in other immune cells like T cells, for example, or eosinophils in their role on infarction. But so far, we are not working on those, or at least we just started to work on those and don't have information that we can share right now.
Okay, and now what type of light sheet microscope from which company, LabVision or Zeiss?
Yeah, that's a LabVision microscope, Ultra Microscope 2. And it's equipped with 4 lasers, single wavelength lasers, so 488. That's in this case for FITS. And 561 we normally use for the autofluorescence of the tissue. And then we have 647 and 785 to get a deeper view inside the tissue. And these laser lines are very good to homogeneously visualize the cells throughout the sample.
The microscope itself has also a 488 laser line, but that is very far into the green spectrum. And at least for the heart, because it's very dense muscle tissue, autofluorescence is quite a problem in these regions, even if we bleach as we do in our protocol. So we only use that for the area at risk determination using FITS, because it's very bright, the fluorophore itself, and it just overshadows the autofluorescence of the tissue. But for single cell identification, we would not recommend to use this far into the green spectrum.
Maybe that's also a good thing to add, because we needed the homogenization of the autofluorescence to use, for example, the Imaris spot function, because that relies on a threshold that you put into. And if you have an inhomogeneous image of your sample, then you don't detect, for example, cells that are in the middle of the sample, whereas you detect others that are outside that are not really cells. And this can only be really assured with the 647 and 785.
Thank you. Now, recently there have been several studies using lectins to label vasculature. Do you see any advantage using CD31 antibody as compared to lectins?
Yeah, we also had a discussion about that before we did the CD31 and everything staining. At least from our point of view, we use very low concentrations of antibody. I think we inject 5 micrograms per mouse, so that's like a 40th part of the antibody usually shipped. And when we search for literature for lectins, we found that they use very high numbers of a very high milligram of lectins. And concerning the costs here, we found actually the CD31 antibody approach to be more cost effective. And but yeah, you can simply use lectins to label the vasculature and image them in the light sheet. That would be possible.
Thank you, gentlemen. Now, we have so many questions that come in, but we're almost out of time, so we're going to have to wrap with this final question. Now, Bill sends in a question. His comment is CD31 is also expressed by leukocytes and platelets at 10 times lower levels than endothelial cells. Also, granulation tissue consists of capillaries, fibroblasts, and myeloid cells and is the earliest stage of healing. The capillary network is very disordered early on, and so perhaps, is this what makes it curly?
Yes, so that's also what we think. So it is, we think it's regrowth, and therefore it's curly and completely disorganized. And also to the first part of the question, can you remind me what that was?
The thing about the
yes. CD31 is also expressed by leukocytes and platelets at 10 times the lower levels than endothelium.
Yeah, thank you. So exactly, it is expressed, but it's so low that we can't distinguish it from the autofluorescence basically in the light sheet. So in the light sheet, you only see the very strong signals at a cellular resolution, but you really need a strong emission in order to detect it properly. And also we place, of course, thresholds for detection over it, and that might also cut away every other expressions that are on a lower basis.
So we had in our studies we never had the problem that positive signals for the capillary network or for the veins and the arteries was also on the same intensity level than that for example, our neutrophils that we tested. It was always we never saw leukocytes with an expression of CD31 that high.
Thank you, gentlemen. Simon and Sebastian, do you have any final comments for our audience?
Yeah, sure. So in general, I thank you guys from LabRoots and Merz for hosting this, and I thank all of the audience for participating so well. So we got a lot of questions and we couldn't answer them all. So, but we will do our job and come back to you and try to answer all your questions later after this webinar. So thanks a lot for your participation and your attention.
Yeah, also from my side, thank you for being here and thank you for your interest in our work. We will try to answer later on everything you might have in store for us.
Thank you, Sebastian. Thank you, Simon. And before we go, I would like to also thank the audience for joining us today and their interesting questions. We had so many questions come in that we are not able to get to. So Simon and Sebastian will answer those questions via the contact information you provided at the time of registration. Again, I would like to thank Simon Merz and Sebastian Kostek for their time today and their important research. We'd also like to thank LabRoots and our sponsor, Merz, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So that's all for now, and we thank you for joining us. Goodbye from California and from Essen, Germany. Have a great day.
Bye.